Hello, good morning everyone. This is Dr. Shyma. We have already started with special senses. I think by now gustation and olfaction would have been over. Now we go to the very major part of special senses, that is vision. And please remember, you need to really concentrate on central nervous system and special senses, because out of 100, almost 60 marks come from special senses and CNS together. So vision basically, it's not a 1 hour class. I'll be doing it in might be 2 or 3 to 4 classes. So in today's class, I'll be giving you all an idea about the functional anatomy of the eye. And later on in the coming up classes, we would go in detail to the image forming mechanisms, the visual pathway, the photoreceptor mechanism, the light and the dark adaptations, and also color vision. So basically, to understand the coming up classes, you need to be really thorough with the functional anatomy that is being discussed in this class. So as usual, I remind you all to take down lecture notes. So basically, if you look at eye, it's one of the most essential sense organs of the living species. Now vision is possible in the presence of light. And accordingly, the eye has been divided into two major parts: an optical system, which actually helps us to focus and form an image on the receptor cells when the light rays are reflected from an object and it falls back on the eye. Now that's the first component of the eye. Now the second component is a neural component, which actually transmits this optical signal in the form of action potential along the optic nerve directly to the visual cortex, where it will be perceived as a visual sensation. Let's go to slide 2. Now slide 2 gives you all a detailed picture of the structure of the eye. And now when you look at this picture, there's a picture of the eyeball. Now the eyeball is a fluid filled spherical organ with a diameter of around 24 millimeters. Now all the optical systems are present in its anterior part, and the posterior part has got the visual receptors. Now from the posterior part, the optic nerve arises and proceeds towards the occipital cortex, where the image is perceived as a visual sensation. Now each eyeball is surrounded by a cushion of fat and is placed within a bony cavity called as the orbit. And if you look at more closely at this picture, you can see a red line and a blue line drawn. And the red line, you call it as the optical axis, and the blue line is the visual axis. Now let's see what this optical axis is. Optical axis passes through the anterior and the posterior pole of the eye. Center of curvature of the cornea and the optic center of the lens are in the same optical axis. Now the blue line, the visual axis, it passes through the cornea but a little nasal to the anterior pole, and posteriorly it passes through fovea centralis, lateral to the posterior pole. We go to the next slide, where you can see that the structure of the eyeball is given. Now the structure of the eyeball, just a second, yeah. So basically in the structure of the eyeball, you can see that the outermost layer is transparent, and its anterior part is called the cornea, and the rest opaque area we call it as the sclera. Sorry, the slide is tilted. Yeah, we continue. So basically the eyeball has got three coats: the outer connective tissue layer, the uveal tract or the middle vascular layer, and the inner neural or the retinal layer. So basically the outermost layer is transparent in its anterior part, and we call it as the cornea, and the rest of the anterior outermost layer is opaque, and it's called as the sclera. The middle vascular layer and the pigmented uveal tract forms choroid and ciliary body in its posterior part, and iris in its anterior part. Now the innermost layer is called the retina, which contains the photoreceptors in association with a neuronal network that is absent in the anterior segment. So now about these three different layers we'll be going in detail. So I think this slide I have already explained to you all what the optic axis is and what the visual axis is. Now this slide, I think this is a fifth slide, and this actually shows you a cross section of the retina, and I think this figure we'll discuss later on in the chapter, where I'll be talking about the innermost layer of the retina. So before that, I think we'll go in detail to the outermost layer, which includes the cornea and the sclera, the middle vascular layer which contains the choroid, the ciliary body and the iris, and then we'll come to the retina. So slide 6, if you look at the outermost layer which contains, one of the component is sclera. Sclera is basically, it occupies the posterior 5/6 of the outer coat is the sclera. It's covered by delicate mucous membrane called as the conjunctiva, and it's made up of fibrous and elastic layers. Now let's see what the functions of sclera is. It offers support and protection. The eye muscles are attached to the sclera. Perforated posterior lamina fibrosa to allow passage of the optic nerve. So basically what the third point I have really briefed up so that it's easy for you all to remember. What the third point tells is the posterior part is perforated, and it's called as lamina fibrosa, which allows the passage of the optic nerve to it. That's what the third point is read as. So we go to the next slide where it's a picture of the cornea. So about the cornea, both sclera and cornea is included as the outermost layer of the eyeball. The cornea occupies anterior 1/6 of the outermost layer. It's a transparent cornea. Cornea is basically avascular and it's highly transparent. And cornea is divided into 5 layers: the squamous epithelium, the anterior limiting membrane of Bowman, substantia propria, posterior limiting membrane of decimates and endothelium. And cornea derives its nutrition from aqueous humor, perilimbal capillaries, and also oxygen from air. Cornea is highly refractive in nature, and the refractive power is around 43 diopter. So now the cornea is transparent. This is a very important point you need to note down. The transparency of the cornea is due to 3 things. Because it's avascular in nature, the stromal fibers that are packed in parallel rows are of uniform diameter, active mechanisms that maintain water content at 77%. These are the 3 reasons why cornea remains transparent. Now when we go to the next slide, the 9th slide, so we have finished with the outermost layer, which includes the sclera and the cornea. Now we come to the middle layer, which is the vascular layer of choroid, which includes ciliary body and also the iris. Now the vascular layer of the choroid, unlike the cornea which is avascular, the middle layer contains blood vessels, connective tissue and also pigments. Choroid forms a posterior 5/6 of the vascular coat, and anteriorly it's limited by ora serrata. Posteriorly it is limited by the entry of the optic nerve. An anterior extension of the choroid we call as the ciliary body. Now ciliary body has got 3 parts: the orbicularis ciliaris, the ciliary processes and the ciliary muscle. Ciliary processes are basically around 70 radial fringes which produces the very important fluid, the aqueous humor. We'll go in detail about aqueous humor. Ciliary muscle contraction produces relaxation of suspensory ligaments, increases anterior curvature of the lens. That's about the vascular layer of the choroid. Now in the next picture, you can see the picture of choroid and the ciliary body. I think whatever the previous slide it explains, you can just look at the picture and try to correlate the parts of the choroid. Now the next, I told you the middle vascular layer contains choroid, which already the details of choroid I have finished explaining. Now we go to iris and also the ciliary body. The iris, the anterior continuation of the ciliary body is iris. It's a vascular pigmented layer, forms a diaphragm in front of the lens. The central aperture is called the pupil. Now there are two types of smooth muscle fibers present here. The circular sphincter pupillae, which is supplied by the oculomotor nerve. Contraction produces miosis. Now the second smooth muscle fiber is a radial dilator pupillae, which is supplied by the sympathetic fibers. Contraction produces mydriasis. Color of the eye is decided by the color of the pigment present in the iris. Please keep that point in mind. Now again, when you go to the next slide, you can see the picture of ciliary body and iris. Now you can correlate the previous slide, what I have explained, with this picture, so that you get a better clarity. Now to brief up the functions of iris, it basically acts as a diaphragm. Now what does it do? Acting as a diaphragm, it controls the amount of light that enters the eye. It helps in focusing of the near object. It acts as a screen, correction of spherical and chromatic aberration of the lens by acting as a screen. That's what the second part of the sentence means. We'll be going into detail of all that spherical and chromatic aberrations in the coming up classes. So that's about the functions of iris. Now the next part, you have the lens. So if you look at the human lens, it's biconvex. The posterior surface has more curvature. The diameter is around 11 millimeters and thickness around 3.6 to 3.9 millimeters at the center. Now the center of the anterior surface will coincide with the center of the pupil. It's made up of flattened epithelial cells that originate at the equator. Now the lens continues. The adjacent margins of the epithelial cells form sutures which radiate from the center. It's surrounded by a capsule which is formed by the epithelial cells present on the anterior surface. Lens is avascular and depends on the aqueous humor for nutrition, and it uses glucose anaerobically. That's a very important point to be noted there. So for its nutritional needs, lens depends on aqueous humor and glucose is used anaerobically. Now about the refractory power of the lens: 16 diopter for distant vision, 28D for near vision, and the disorders associated with the lens is presbyopia and cataract. Now about all this we'll be doing in detail in the coming up classes. This is just giving you all a brief introduction. Now we go to the formation of aqueous humor. Now aqueous humor alone can be asked as a 5 marker for you all. Now if you look at aqueous humor, with the diagram you'll have to make a note of the points which I'm going to tell now. Have a closer look at the figure. Aqueous humor is basically a protein free clear fluid from which the lens and the cornea derive their metabolic requirements. Now in the ciliary processes, it is formed from plasma by the mechanism of ultrafiltration, active transport and diffusion. And the composition of aqueous humor is very similar to that of plasma, but it lacks proteins and has very high concentrations of ascorbic acid compared to plasma. Now the ciliary process form aqueous humor and pour it into the posterior chamber, from where it flows through the pupil into the anterior chamber. That's a circulation of aqueous humor. So that particular thing what I've told now, you can actually see it in the figure. Now the ciliary process form the aqueous humor and they pour it into the posterior chamber, from where it will flow through the pupil into the anterior chamber. Now you need to know, what this posterior chamber is, what this anterior chamber is. Sorry. Now posterior chamber is basically a triangular space enclosed anteriorly by the back of the iris, and posteriorly it is limited by the anterior surface of the lens. Now laterally, its base is formed by the ciliary body. Medially, the apex is situated at the point where the pupillary margin comes in contact with the lens. So that's about the posterior chamber, the extent of the posterior chamber: anterior, posterior, medial and lateral. Now let's see what the anterior chamber is. Anterior chamber is bounded anteriorly by the posterior surface of the cornea and posteriorly by the anterior surface of the iris, and the anterior surface of the lens that is not covered by the iris. So basically, I'll just repeat it. Anteriorly, it's bound by the posterior surface of the cornea, and posteriorly there are two boundaries being set here, by the anterior surface of the iris and also by the anterior surface of the lens, which is not being covered by the iris. Now both these posterior chamber and the anterior chamber are filled with aqueous humor, and the aqueous humor pressure is about 15 to 18 millimeters of mercury, which is higher than the intracranial pressure. Please keep that in mind. Basically, this pressure helps to maintain the shape of the eye and keeps the vitreous humor pressurized. Now from the anterior chamber, the aqueous humor gets absorbed by the spongy meshwork of trabecular tissue, and it's drained into the canal of Schlemm. Canal of Schlemm is basically a venous channel at the angle of anterior chamber. Now the anterior chamber is a junction between the iris and the cornea. Now basically the obstruction to this passage will result in very high intraocular pressure, causing glaucoma. The aqueous humor is continuously formed from plasma, and a complete turnover occurs in about just 1 hour. Okay, so that gives you about what aqueous humor is, how it is formed, how it is circulated, and when occurs a block or an obstruction in the passage, how it results in glaucoma. The whole thing what I've explained now, if you can take down notes, that completes your 5 marker. So now I've just briefed up what I've told you on now. It's formed by the ciliary processes, by active secretion and also by ultrafiltration, and about 2 to 3 cubic millimeters per minute is the rate of formation. It fills both the anterior chamber and the posterior chamber. pH is around 7.1 to 7.3. The referral index is 1.13. Composition is less glucose and then plasma with a high concentration of ascorbic acid. I think circulation of aqueous humor I have already told you all, and here I have mentioned it in point wise so that it becomes easy for you all to remember. And in this slide you can see a picture which will actually give you all more clarity on understanding the route of circulation of aqueous humor. Now if you look at the functions of aqueous humor, it provides nutrition to the cornea and the lens. It maintains the intraocular pressure. It maintains the shape of eyeball, and it also acts as a refractive medium. Now about the intraocular pressure, because you know that intraocular pressure is maintained by the aqueous humor. So now intraocular pressure can come as a 3-marker question for you all. The normal intraocular pressure is 10 to 20 millimeters of mercury, and it's measured by a tonometer. Level of pressure is determined by the resistance to flow at the trabeculae. Now the intraocular pressure, when it's greater than normal, we call it as glaucoma, and there are 2 types of glaucoma: the closed or the narrow angle glaucoma, and also the open angle glaucoma. Now let's see what the closed angle glaucoma is, and glaucoma can come as a 5-marker or a 3-marker for you all. Now the closed angle glaucoma occurs in women in 5 to 6 decade age group, that means in the age group of 50 to 60, and it prevents the outflow through the filtration angle. Now in closed angle glaucoma, the symptoms are blurring of vision, the halos around the light, and headache. Acute type of closed angle glaucoma, intense pain with vomiting, and vision is also reduced here. Now if you look at open angle glaucoma, it affects either sex in the 7th decade, that means in the age group of 70s, due to vascular sclerosis or sclerosis of drainage channels, headache, and field defects are common with open angle glaucoma. Now the effects of glaucoma, if you see, the cupping of optic disc, field defects are present, blindness if pressure is continuously elevated. These are, the three effects of glaucoma, the most important ones which have been summed for you all. Now if you look at the treatment, the medical treatment involves pilocarpine, adrenaline and acetozolamide. Surgical intervention will be by trabeculectomy or iridectomy to facilitate drainage. Now about the vitreous humor or the vitreous body. One second, let me just go to the, okay fine, yeah. So the vitreous humor, the vitreous body, it's all the same. It's basically a transparent avascular tissue which has got the consistency of gelatin and which occupies the posterior cavity of the eye. Now this jelly-like mass, it contains loose collagenous fibers and vitrine. And this vitrine is a highly hygroscopic protein which is formed much early during embryonic life and it doesn't get replaced thereafter. Now the portion of the vitreous which is present behind the posterior surface of the lens acts as a cushion for the lens. Now in the fetal life, the hyaloid artery runs anterior-posteriorly in the middle of the vitreous and it nourishes the vitreous and also the fibrovascular sheath of the lens. Now in the later months of the intrauterine life, the artery gets obliterated and immediately after birth, it disappears, which leaves an empty narrow channel which is known as the hyaloid canal. If the hyaloid artery persists, basically it becomes a congenital anomaly which causes defective vision. The avascular vitreous may show degenerative changes in old age and high myopia, in which cases it loses its gel-like consistency and gets liquefied. So here, very few points I have briefed down about the vitreous body in this slide. It's a transparent hydrophilic jelly. It lies behind lens and the ciliary processes. Light rays passes through the vitreous to reach the retina. But these 3 points basically, are the most important points which you can sum down if vitreous humor comes as a 3 marker. But if vitreous humor is asked as a 5 marker, you will have to really pen down, the points from whatever I have told you all, explained to you all now. Now, if you look at the refractive media of the eye, what you should understand is light travels or traverses would be a better word through several media in the eye to reach the retina. Now the incident light ray travels from air to pass through several transparent media before it's finally going to touch the retina. Now it passes through a thin film of tears, cornea, aqueous humor, lens and the vitreous humor. So this slide where it talks about the refractive media of the eye, refraction occurs at the interface between the refractive media. Cornea has a refractive index of 1.37, aqueous humor 1.33, lens 1.42, vitreous body 1.33. Now this particular figure, this data we'll be doing in detail when I talk about refraction and how the image is formed process. You will have a better understanding of the concept of refraction which occurs at the interface between the refractive media as the incident light passes through the different structures of your eye before it touches the retina. Now this part is really important for you all, the layers of the retina. It can come as a 5 marker. Now if you look at the layers of the retina, I think I would like to go by this figure. This figure is very important. Now here, so you have around 10 layers here. So I would just like to enlarge this figure. So we start from the pigment epithelium. Then you have the rods and the cones, which is divided into the outer segments of the rods and the cones and the inner segments of rods and the cones. Then you have the outer nuclear layer, the outer plexiform layer, the inner nuclear layer, the inner plexiform layer, the ganglion cell layer, Finally, you can see the optic nerve fibers taking the sensations to the visual cortex. So this is basically the layers of the retina. I'll take you all to the slide once again. So we just repeat the different layers of the retina: the pigmented epithelial layer, the receptor cell layer, the rods and the cones, the outer limiting membrane, the outer nuclear membrane, the outer plexiform layer, the inner nuclear layer, the inner plexiform layer, the ganglion layer, and the inner limiting membrane. So you need to mention these layers with this figure. You can actually find, little difference between these names and what is given in this picture, but it's one and all the same. And so I would recommend you all to list down, because anyway, while mentioning the layers of retina, you will have to draw this figure. So it will be better if you can, just take down and write these, the names of different layers of the retina, which is given on, the left side of the figure. So you can just draw this figure and put it on the left side. You need not really waste time taking it out and again penning it down on your paper. Now it comes, so basically now what happens is you need to I've put it in different slides, but I think I would like to explain what each one is from this figure, so your clarity is more. So now let's see about the pigment epithelium. Now the pigment epithelium basically is a single row of cuboidal epithelium, and it contains melanin. Melanin is a pigment, and this pigment epithelium, it prevents the scattering of light and reflection by absorbing excess amount of light. So that's what the role of pigment epithelium is. It's a single row of cuboidal epithelial cells, and it contains melanin pigment. Now what is the function of pigment epithelium? It will prevent the scattering of light and also the reflection of light by absorbing the excess amount of light which is falling on the retina. Now coming to the next layer, you have the layer of rods and cones, which contains peripheral processes of the receptor. I think you just need to tell only that one sentence about the layer of rods and cones. Now the third layer, you have the outer nuclear layer, which contains the cell bodies of the rods and cones. So I think where were we? We were at the outer nuclear layer. Yeah, correct. The third one, which contains the cell bodies of the rods and cones. And then you have the fourth layer, which is the outer plexiform layer, which is basically the synaptic zone of central processes of receptors with peripheral processes of bipolar cells. Now the horizontal cells make synaptic connection with the receptor and the bipolar cells, and they modify their activity by lateral inhibition. Now what this lateral inhibition is and all, you will get a better clarity when we do central nervous system immediately after special senses is over. For time being, I think if you can just memorize those many points I have told you all about the different cell layers, that's more than enough, and that will make up to a 5 marker if retina is asked. So I think all that what I have told now, I have appended down in points under pigment epithelium, layers of rods and cones, the outer nuclear layer, the outer plexiform layer, inner nuclear layer, inner plexiform layer, the ganglion cell layer. I think this is the same, a picture, where the light falls and, in darkness how it looks like. I think we can just skip these figures. And here basically it talks about a little more about the rods and cones. Rods and cones are basically the photoreceptors present in the eye, and it consists of peripheral processes, cell body, and the synaptic zone. You can see that very clearly in that picture of the layers of the retina. It distributed more in number towards the posterior pole, and you can see that both rods and cones are absent over the optic disc. Now at the fovea centralis, only cones are present, and rods mainly are present in the periphery, and you can see that the cones are more concentrated in the central region. Now this is a picture of the rods and the cones, and about the rods, there are 120 million in number. They are rod shaped, slender and elongated, and they contain a pigment called as rhodopsin. Their receptive field is more for rods, resolution is less, and rods are responsible for the scotopic vision, and loss of rod function leads to nyctalopia. Now if you look at cones, their number is 1.6 million, 120 million, so you can compare the number. Concentrated in the central retina, they are conical in shape, that's why they get the name. less pigment are present per cone. And the cones are responsible for color vision, photopic vision, and their resolution is more, 1:1 at the fovea. Loss of cone function leads to functional blindness. So both rods and cones can come as a 3 marker, and please remember that you don't skip any points, because I have penned the maximum important points. Now don't try to cut short from the contents what I have made a note in the figure. Now if you look at the functions of retina, the light, it can sense light, color sense, form sense, and the sense of contrast. Now the reflexes, the retina takes care of us, the light reflexes, the accommodation reflexes, and the viscospinal reflexes. Now about all this, I'll be doing it in detail in the coming up classes. Now the retinal impulses are required for tone, posture, and equilibrium. Now the circadian rhythm is also a very important function of the retina. And when you look at this picture, you can see that the retina, which now this picture is basically a fundoscopic view of the retina. You can see the optic nerve and the fovea in the picture very clearly here. Now, if you look at the protective mechanisms of the eye, it's been listed down in 3 points. The bony walls of the orbit, the eyelids, which is protective, closure, isn't it? Now, the reflexes are the dazzle reflex, the menace reflex, and the lacrimal gland, which produces tears, is also, one of the protective mechanisms of the eye. So, the bony wall of the orbit, the eyelids, which takes care of the dazzle reflex and the menace reflex, and the lacrimal gland proves as protective mechanisms of the eye. So, thank you. There we, I come to an end of, the introductory class on vision, where I have only given you all an idea about the functional anatomy of the eye. So before I actually go to my next class tomorrow, I want you all to be very thorough with this, so that when I go to the actual mechanism of how you see an object, what accommodation reflexes, what light reflexes, I would want the base to be very strong. So please make down notes from whatever the extra points I've given you all during my lecture. Make a mind map and be thorough with the contents, so that in the coming up 2-3 classes is going to be really great. So thank you guys for listening.